Now, I probably confused you with turning genes on and turning genes off. But the reason that I did that is that there are two immune cells that are very important in the context of MS. One we call a Th1 cell, a helper type 1 cell, and one we call a helper type 17 cell. Type 1 because it makes a compound called gamma interferon, and gamma interferon in this context, in the context of MS, is bad, and interleukin 17, which is the 17, and all these cytokines are called interleukins, and they're numbered sequentially as they're discovered. So we're now up to 37. So we've exceeded the Baskin-Robbins number. But in these two kinds of cells are believed to be the important players in the cells that can damage MS. So in MS. So that recognition process that we've talked about, that amplification process, ends up producing these cytokines or proteins that cause inflammation that can damage tissues. So that's what this process is all about. And that's the IL-17s. Now, what happens in the production of cytokines is that cells again interact with one another. We've talked about this over and over again. These are the cells that may pick up, let's say, a mycobacterium or a measles virus or whatever, and they instruct these cells that are helper cells that make the cytokines, either gamma interferon or 17 in this case, which then go ahead, and in this case is just an example of cells that are killer cells, will activate these cells, which then go ahead and do their business. Or the helper cells keep interacting with the macrophage or dendritic cell, make that more active so that when that interacts with the cell that's going to be the killer cell, it amplifies the process. So these cytokines that are damaging in MS, not only in and of themselves can cause tissue injury, but they're part of the amplification process, which adds insult to injury. And that's what really happens in this process. Now, just as an example, again, to show you how cells work, these are cytolytic T cells here. You can see they latch onto a target cell that, for example, here may have a virus, but may just, as an example, also be a myelin cell, the oligodendrocyte. In the cytolytic cells, they have these little packets we call with enzymes, and they actually inject them into the target cell over there. And this over here is just a molecular way showing the molecules at the surface that bind them, and the blue over here are these packets that are going to get injected into the other cell. So again, these are cells that can be harmful and damaging and lead essentially to the destruction of that cell. And it can either be due to the cells adhering to one another or just to those cytokines that are floating around and latch onto a cell and then start causing damage. And both of these processes, to a greater or lesser extent, are what happens to the tissue myelin in the case of MS. So what actually happens then is the following, that we have cells that circulate through the body that may, when they're activated, latch on to the inside of the cell that we've talked about in the context of Tysabri, go into the tissue over here and cause a problem. And if you remember again with Tysabri, there's that sticking process that occurs that gets interrupted over there by antibody which is bound onto the cell. So the antibody would bind on here, and because it's there, it blocks the interaction between one molecule and another. So again, that process we know is very important in the case of MS. And in the case of MS, when the sticking process occurs, if you're having an MRI and you're given an intravenous contrast injection, the contrast, and this is the inside of the blood vessel over here, just enlarged, the contrast normally should stay in the blood vessel, but in fact it leaks through because this process is going on. So when you get MRIs, the reason you get them not only without enhancement but with enhancement is to try to determine if this process over here is actively occurring at the time you get the injection. Okay, and we know by inference that when we see the gadolinium leaking out, that this is the process that's actually going on from an immune point of view. So the bottom line of all of this is that we have this whole process of recognition of an antigen in a tissue, triggering an immune response, activating an immune response, getting the cells there, and the cells come in and then can cause damage. The immune system is also set up to end the reaction that's occurring. And we call that suppression, and it's called by regulatory T cells. That's a new fancy name for what used to be called suppressor T cells. So basically, the immune system also learns at the other side to downregulate the response. And whether or not you develop MS or any other autoimmune disease is really a balance between cells that can potentially cause damage and cells that are there to counteract those cells to try to settle it down and prevent it from happening, or if it starts happening, to try to abort it and prevent it from going on. So these, and in the case of MS or other autoimmune diseases, the balance is lost, and so the balance is shifted towards cells that potentially can cause inflammation and damage, compared with the cells that normally would settle things down or keep it under wraps to begin with. This again just emphasizes the same thing, that these cells get activated, and that let's not go there and skip over that. So for example, interleukin-17 that I told you was bad is something that's produced primarily in response to bacteria. It gets activated, the cells produce it, and they can go ahead and cause inflammation, which then causes injury. The irony of the immune system is that the very cells that can cause damage in and of themselves can make other cytokines which actually may promote healing or reduce injury of the immune system. So even within the cells that can cause damage, there's this fail-safe mechanism to try to put a stop to it. It allows it only to go so far, but no further. So there are layers on layers on layers of checks and balances that occur in the immune system, which makes it basically difficult to occur, but when it occurs, we know that it unfortunately goes ahead and causes damage.